Hey there, science fans. Today, we're diving into the amazing world of DNA and how it makes you, you. Think of your DNA like a giant library full of blueprints. These blueprints contain all the instructions to build and run your body. This set of instructions is called the genome, but there's a catch. These blueprints are written in a secret code locked away inside the nucleus of your cells. So, how does your body actually use this information? That's where the central dogma of molecular biology comes in. This dogma explains how we go from DNA to the proteins that make us tick. First, the DNA blueprint is copied into a messenger molecule called RNA in a process called transcription. Think of RNA as a temporary copy of a single recipe from our blueprint library. This copy can then leave the nucleus and head to the ribosomes, the protein factories of the cell. It's at the ribosomes where the RNA message is used to build proteins through a process called translation. Pretty cool, right? Now, let's unravel the mystery of DNA transcription together. Imagine your DNA as a cookbook, filled with incredible recipes for delicious meals. But you can't take the whole cookbook into the kitchen, right? That's where transcription comes in. Transcription is like carefully copying a single recipe from your DNA cookbook onto an index card, which represents the messenger RNA or mRNA. This way, you can take the recipe to your kitchen counter, the ribosome, and whip up a tasty dish, a protein. This process of copying the DNA recipe onto mRNA is crucial because DNA is too precious to leave the nucleus. Transcription ensures that the genetic information is delivered safely to the ribosomes without damaging the original DNA blueprint. Now, let's break down transcription into 3 easy steps: initiation, elongation, and termination. First up is initiation, like pushing the start button on our recipe copying process. For transcription to begin, an enzyme called RNA polymerase needs to find the beginning of our recipe or gene. It does this by searching for specific regions on the DNA called promoters. Think of promoters like recipe titles in our cookbook that tell RNA polymerase where to start copying. Once RNA polymerase finds the promoter, it attaches to the DNA and starts unwinding the double helix, like unzipping a zipper to reveal the DNA sequence that needs to be copied. With RNA polymerase bound to the DNA and the recipe open and ready, the process of transcription can begin. Building the message elongation. With initiation complete, it's time for elongation, where we start building our mRNA molecule one nucleotide at a time. As RNA polymerase moves along the DNA strand, it reads the DNA sequence and adds complementary RNA nucleotides, the building blocks of RNA, to create a growing mRNA chain. It's like meticulously copying each ingredient from our DNA recipe onto our mRNA index card. Remember, RNA has uracil instead of thymine, so whenever RNA polymerase sees an adenine on the DNA, it adds a uracil to the mRNA strand. This process continues with RNA polymerase adding one nucleotide after another, creating a longer and longer mRNA molecule that carries the genetic instructions from the DNA. Wrapping up, termination. Just like every good recipe has an end, so does transcription. This final step, called termination, is how we tell RNA polymerase to stop copying. As RNA polymerase moves down the DNA, it eventually encounters a specific sequence that signals the end of the gene, kind of like the end note at the bottom of our recipe. This sequence tells RNA polymerase to detach from the DNA and release the newly synthesized mRNA molecule. Now, our mRNA molecule carrying the precious genetic instructions is free to leave the nucleus and head to the ribosome. It's time to move from recipe copying to actual cooking. From recipe to masterpiece, RNA processing and beyond. But wait, there's more. Before our mRNA can be used to make proteins, it needs to undergo some important processing. First, a protective cap is added to the beginning of the mRNA, called the 5' cap. Then a tail of adenine nucleotides, called the poly-A tail, is added to the end. Finally, the mRNA undergoes splicing, removing introns and joining exons. Now, our fully processed mRNA is ready to leave the nucleus and head to the ribosome. This protein will then perform a specific function in the cell. So next time you think about the amazing complexity of life, remember the journey from DNA to RNA to protein. Keep those questions coming and never stop exploring the amazing world of science.